All right, so this is going to be the second topic in biochemistry. Now, these topics you can see are long, but then what can happen is that, like for the previous academic year, you'd be in class for one course for 4 hours. And I remember one time we learned anatomy from 9 hours to 20 hours, one course. So be prepared to learn. So this is carbohydrates. I have some knowledge already from first year, of course, but there is nothing that is to be taken for granted at any point. So carbohydrates, you'll find them in plants and in animals that we know, and they are the most abundant biomolecule, not just in humans but on Earth. You know that for biomolecules, these are molecules of life, and the ones we talk about are carbohydrates, lipids, proteins, and nucleic acids, enzymes. Those are the biomolecules we talk about. But these ones, the carbohydrates, are the most abundant. Okay. So carbohydrates, you know that when you are writing from the structure of a carbohydrate, I think we've been doing that from first year. You saw that they had OHs. Okay. So they are polyhydroxy, meaning they have got a lot of OH groups. They can either be aldehydes or ketones. So depending with where they are going to be aldehydes or ketones, that will also determine their name. The question should be, what is the difference between a carbohydrate and a ketone? Okay. Now, when you're talking about these carbohydrates and a ketone, they all have a carbonyl functional group. Carbon double bonded to oxygen. That is what is known as a carbonyl. Now, when this carbonyl group is at the end of the chain, let's say for example, you had something like this, and then you have it at the end of the chain. This is known as an aldehyde. When the carbonyl group is on the middle, any position as long as it's not at the end of the chain, that becomes a ketone. So this is how these guys are. They have got a lot of hydroxides, meaning you see them like this with a lot of OH groups. Now it's taking a bit of time for it to reflect if I write, so you bear with me. Okay, so this is how these guys are going to be. So you can see they have got a lot of OHs, that is a polyhydroxy. But for this one, you can see the carbonyl group is at the end of the chain, meaning this one is going to be an aldehyde. Most carbohydrates are going to have the empirical formula CH2N and CH2O with N. You can also write it as CN, H2N, and then ON, but not O. This formula is generally for monosaccharides. Okay, but even for the monosaccharides, is not all the monosaccharides. Okay, carbohydrates are going to be classified based on their size. So on their size, you are going to classify them as either monosaccharides, disaccharides, oligosaccharides, or polysaccharides. Depending on their functional group, you can classify them as aldehydes or ketones. Right? Now, what are the functions of carbohydrates? Carbohydrates can be used as that is staples, and the oxidation of carbohydrates is very key in the yielding of energy, the formation of energy. Now, you've always been talking about carbohydrates form energy from high school. You are now going to understand how do they really form energy. We are going to do that as a topic station, all right? So, apart from being used as staple and also as yielding energy, carbohydrates can function in structural carbohydrates. Now, these ones which function in structures, they are used for protection in the cell wall of bacteria and plants. Okay, they can also be found in connective tissue. Now, when we say connective tissue, what we mean is this. Look at this. When you have got one brick, you want to form a house, and then you have got another brick. If you just stack these bricks together, you are not going to have a house at the end of it all. So, what are you supposed to do then? You need to also have some other things, cement to be in place. You also need water. So, those things that you need to hold the bricks, that is connective tissue. So, in the body, connective tissue is used for a lot of things, and we are going to do this one in anatomy. It's a topic on its own. Okay, connective tissue. So, the body is made up of cells. It is also made up of connective tissue because those cells have to be connected together. For you to connect them, you need also connective tissue. It's not the only tissue that is there, also epithelial tissue and other tissues come in play. But we need to know that carbohydrates are used in structures for protection, bacteria in the cell wall. Okay. Carbohydrates are also used to lubricate skeletal joints. So, for example, on your elbow, you need carbohydrates for lubrication there. Other carbohydrates are going to be used to act in cell to cell recognition and adhesion. Let's say, as I've said, you have got one cell in the body and another cell. How does this cell hold to another? So you are going to have carbohydrates being used in that. Also, cell to cell recognition. For a cell to recognize that this other cell is part of the body, carbohydrates are important. Apart from that, carbohydrates are going to be important in blood clotting and also in immune response. So those are some of the functions of the carbohydrates. Okay, are we good? So be free to ask if you have got any question at any point. Now let's start talking about the classifications of these carbohydrates. The first ones are the monosaccharides. Monosaccharides are simple sugars. They only have a single polyhydroxyaldehyde or a ketone, only a single one. Okay, meaning you don't connect these polyhydroxyaldehydes or ketone. If you have got one aldehyde, it's just one ketone, only one. So the most abundant you need to know is the D. The most abundant monosaccharide is D glucose. It's the most abundant. It is also referred to sometimes as dextrose. Okay. Now, these monosaccharides can be classified depending on the number of carbon atoms. So, when you classify them depending on the number of carbon atoms, they can be classified as trioses, tetroses, pentoses, hexoses, heptoses, depending with how many carbons they have. But you can also classify them depending with whether they have got an aldehyde or a ketone. And in that way, you are going to classify them as being aldoses or ketoses. Okay. So how many carbon atoms are in a triose? Anyone? Very good. Three carbon atoms. Tetros, four, pentos, five, and just like that. Okay. So the simplest monosaccharide, which we'll talk about, which is of significance biologically, is glyceraldehyde and dihydroxyacetone. Should have learned in first year that glyceraldehyde can exist in the L or D form. So I've got D glyceraldehyde and the L glyceraldehyde. And then the dihydroxyacetone can't exist in those forms. It's only one. Okay. So the glyceraldehyde is also known as aldo triose because it's an aldehyde. Look at the functional groups at the end of the chain, and it's a triose. Wow, dihydroxyacetone is a keto triose because it has got a functional group on the middle. Now you need to understand this because these things will come later on as we'll be discussing. Okay. Monosaccharides of more than 4 carbons, you need to know they can exist in cyclic form. That is because they have got that tendency of bending, and then the anomeric carbon. If we talk of anomeric carbon, we are referring to the carbon that contains the functional group. This carbon will react with the carbon that is second from last. And then what you are going to have is a circle. So they are going to form cyclic structures. Monosaccharides are going to be generally colorless, water soluble, crystalline solids, like salt, the way salt or sugar is. Some of them they are going to have a sweet taste. Okay, some characteristics. So talking about trioses, you can see we have got an aldehyde being glyceraldehyde and a ketone being the hydroxyacetone. Okay, those are trioses. You also have tetroses. Please, you need to know which ones are aldehydes, which one are ketone. Please, you need to know. So for those with 4 carbons, this one you didn't do in first year, but you need to know now. You have got erythros. Erythros is an aldehyde. Now, erythrulose is a ketone. Just remember in first year, we talked about ribose and ribulose. Okay, ribose and ribulose. So, ribose, something that has got just O, is just an aldehyde. If you have got a U, that becomes a ketone. Same with the erythros and the erythrulose. You need to know because they can ask you if you can identify which one is an aldehyde and which one is a ketone. Another aldehyde that has got 4 carbons is threose. Okay. Don't think of three and then you say no, it has got 3 carbons. That's threose, not meaning it has 3 carbons. Okay, so that is on the tetroses. And then we are also having pentoses. Pentoses have got 5 carbons, and some examples on aldehydes we have got ribose, which you already know, arabinose, xylose, and then the lexose. These are aldehydes. You can see basically the same formula. What is just differing is how some atoms are arranged. Now, not really necessary for you to know the way atoms are arranged. All you need to know is at least which one is an aldehyde, which one is a ketone. At least don't forget xylose, sepentose, which is an aldehyde. Okay, ribose, you already know that from first year. On ketones, you have got ribulose, same just like from the previous one where we add erythros and the erythrulose. Okay. You also have xylulose. Just like here we have the xylose. It was an aldehyde. If you put a U somewhere, xylulose, it becomes a ketone. You also have exo sugars. Exo sugars, these guys have got 6 carbons. You can see we have got aldehydes examples: allose, outros, glucose. So these ones which been encircled are the big ones. At least you need to be able to know really. glucose, mannose, and galactose. On the ketones, you have got the secone, fructose, sobose, tagatose. But the most important one on the ketone is fructose. At least you need to know this one, okay? So those are exoses. Another thing that you need to understand about these monosaccharides is that they can exhibit isomerism. Isomers are compounds with the same chemical formula but different structures. Okay. Examples of isomers, you have got fructose, glucose, mannose, galactose. So you can see one is a ketone, the others are aldehydes. Why are they isomers? Because they are having the same chemical formula C6H12O6. But look at their structures. Their structures are different. Those are isomers. Now, you also have what is known as epimers, not only isomers. Hope we are good on isomers. Are we good? Okay. You also have epimers. But what are epimers really? So epimers are carbohydrate isomers that differ in configuration around one specific carbon atom. Only one. Okay. Everything else is going to be the same, but they only differ at one specific carbon. For example, glucose and galactose are epimers. This actually came in last year's exam, and it was writing, not a multiple choice. Okay, write examples of epimers for glucose. So glucose and galactose, they are going to be epimers. They are epimers at what carbon? At carbon number 4. If you see, this is glucose, this is the galactose here. Look at where it is blue. Okay. For glucose, the OH is on the right. For galactose, the OH is on the left. So that is the difference. But for glucose and mannose, they are also epimers, but they are epimers at carbon number 2. For glucose, the OH is still on the left, but for mannose, the OH is on the right. So epimers are not the same as isomers. Of course, they are isomers, yes. But to be specific, if they just differ at one point, they are known as epimers, to be specific. Are we good so far? Okay. Now, we also need to understand enantiomers. Okay, there's a mic which is on. Is it a question or something? Pardon? Okay. All right. So we now want to understand what enantiomers are. Now, enantiomers will really talk about all these we've been talking about in organic chemistry properly. Enantiomers are mirror images of each other. Mirror images. For example, your left hand is a mirror image of the right. So you call those as enantiomers. Okay, enantiomers. So when you're talking about enantiomers in relation to these monosaccharides, you talk about the D and the L form. Those are enantiomers. So for the D form, the OH on the asymmetric carbon, furthest from the carbon is on the right side. Now, when you talk about asymmetric carbon, it's a carbon that is bonded to four different things. Okay, four different things. And the asymmetric carbon, which is furthest from the carbon. So the carbon is this one, or the carbon containing the functional group is the one on top. Asymmetric carbon, all these carbons, carbon number 2, number 3, number 4, number 5, they are bonded to four different things. So the furthest is this one. So you can see if the OH is on the right, that is D. If it is on the left, then that is L. Those are what we call as the enantiomers. So monosaccharides can also exist in cyclic form. Okay, they can exist in cyclic form. So what really happens is that the anomeric carbon, as I said, the carbon that contains the functional group, will react with the second from last carbon. Let's say this is the carbon which contains the functional group. Okay, it's supposed to be in COO. Let's say this is COOH. And then you have got a carbon here, a carbon here, a carbon here, a carbon here, and then at the end you have got CH2OH. So the second from last carbon is this one. So what happens is that the carbon containing the functional group will come and react with the second from last carbon. So if something has got 6 carbons, the second from last carbon will be the fifth. Something has 5 carbons, it will be the fourth. When they do react, they'll now be able to form the circle. Now, alcohols, an alcohol has got OH. These alcohols can react with aldehydes or ketones. In other words, the OH can react with the carbonyl group, which is CO, C double bonded to O. When these react, they can form hemiacetals or hemiketals. Okay, hemiacetals or hemiketals. This is a very important organic reaction, and to understand its mechanism in chemistry is very important, and they will bring it, write the mechanism for the reaction of this and this. So you need to be able to know how it happens. But then that is what you just need to understand, that when an aldehyde or a ketone reaction with an alcohol, they can form. So for an aldehyde with an alcohol to form hemiacetal. A hemiacetal is a compound that has got an OH functional group, and then also with this group. So this group is, we call it an ether. Okay, oxygen bonded to some other group, that's an ether. That is hemiacetal. Now when you get to substitute, when you get to form where the group which was alcohol, you still substitute it again, the hydrogen which was there, you remove it, you again substitute with an other group. So now you no longer have an alcohol, you both have two other groups. So it becomes an acetal. Hemiacetal has got an OH and an ether. Acetal has got two ethers. Now, it's not something to worry about in this course, because there's somewhere where you need to understand it in chemistry, not here. Okay? Okay. So another thing that you need to know is that glucose, specifically the one in the D form, can form a ring and is called a Paranos ring. A Paranos ring is a ring that looks like so. It looks like an hexagon. That is a Paranos ring. Now, it's not all compounds which have got 6 carbons which form a Paranos ring. Fructose has got 6 carbons, but it forms a furanous ring. A furanous ring looks like a what is this shape? A, okay, it's penta. It has got 5 carbons. Okay, pentagon. It looks like a pentagon. So every time something has got a pentagon shape, that is a furanous structure. Even if fructose has got 6 carbons, but it forms a furanous ring. Okay, so that is what you need to take note. Isomeric forms of monosaccharides that differ only in their configuration about the hemiacetal or the hemiketo are called anomers. They are known as anomers. So when they just differ on the configuration of the hemiacetal or the hemiketo. Remember what we said about hemiacetal? When the alcohol just basically react with either your aldehyde or your ketone, you form those compounds. Okay. Now, some interesting things. So these monosaccharides that we have talked about, they can have derivatives. In other words, You can remove either an OH and substitute it with another, or you can oxidize the OH, or you can reduce the OH. By definition, what is oxidation? Oxidation is the gain of oxygen. So meaning if I have a compound which was having a carbonyl functional group. This is a carbonyl functional group. And then we make this carbonyl functional group to become a carboxylic acid functional group like that. What we have basically done is we have oxidized it. So this is oxidation. Okay? Oxidation can also be defined as the loss of hydrogens. So if a compound had an hydrogen and then it loses that hydrogen, then it has undergone oxidation. The opposite is going to be true on reduction. So for you to be able to form a sugar derivative, you can substitute an hydroxyl group with another group, or you oxidize that hydroxyl group to a carbonyl group. That is how you form sugar derivative. The first one you need to understand is aldonic acid. Now these are exam questions. Make sure you understand, and this was there in last year's exam. Aldonic acids, aldonic acid. Now an aldonic acid is going to be formed when the aldehyde group of carbon number 1 is oxidized to a carboxylic acid. Please understand. Look at this. This was an aldehyde group. What was the functional group? It was COH. This group, COH. Look at that. It was COH. It has now become COOH. What did we say? Every time something happens like that, we gain an oxygen. That is known as oxidation. Specifically for aldonic, it is at carbon number 1. For example, glucose can be oxidized at carbon number 1, and then you are going to form gluconic acid, because these are aldonic gluconic acid, right? So gluconic acid is important in application in the food industry, okay, and also in cosmetics as antioxidants, things which prevent oxidation, okay, in cosmetics. So that is some function of gluconic acid. So remember, carbon number 1, it is aldonic. The next one is known as uronic acid. So the first one, remember, we are starting with the letter A. Okay, the letter A. So you can think of it, carbon number 1. This one is the letter U. So it is the last carbonyl group. Okay, actually the last hydroxyl group. So at position number 6, the hydroxyl group is oxidized into a carboxylic acid. So this group here has been oxidized. You can see we gained an oxygen, or we lost hydrogens. Initially there's supposed to be like 2 hydrogens there, but you can see it's now, or there are supposed to be 3 hydrogens down if you can see the structure. But now we only have 1 which has remained. We're supposed to only have 1 oxygen, but we now have gained 1. So that is what we call oxidation. For example, you can get glucose, you oxidize it at carbon number 6, it becomes glucuronic. Glucuronic. So what did we do? We just got from the word glucose, we remove the SE at the end, and then we actually removed the OSE at the end, and then we put the word uronic. Became glucuronic acid. Different from aldonic acid. What did we do? From glucose we removed the OSE, and then we got the last 4 letters of aldonic, became gluconic. You need to know how also, because I can give you an example, and then if you don't identify, you can't identify what happened. Okay, so glycosides, or compounds which are going to come from D-glucuronic acid, and other steroids. Now, steroids, these are compounds which are made up of cholesterol. Cholesterol is like a lipid which has got four chains. These guys are going to appear in urine of animals as metabolic products. So foreign toxic substances are going to be converted into glucuronides, which are basically some different forms of glucuronic acid. You are going to, if you remember, you come across this term as we'll be talking about some other topics in front, like the hair metabolism. Okay. So in the, let's say, when you are forming urine, starting also in the liver, foreign toxic substances which are going to go to the liver will be first converted to glucuronides before they can be excreted into urine. That is known as conjugation, and this is a very important process that we are going to we'll discuss. This is a very important process. Okay, so D-glucuronic acid is a major component of connective tissue. We already talked about what connective tissue is, things which hold cells and such. Okay, so these are uronic acids. The other type of derivative is aldaric. Please, this is not aldonic. The first one was aldonic, then we went to uronic. This is aldaric. There is no O, there is A. So aldaric acids are formed when both carbon number 1 OH and carbon number 6 OH have been oxidized to carbonyl groups. Okay? In other words, it's a combination of aldonic acid and uronic acid. For example, these are aldaric acids. Glucose can be converted into glucaric acid. What did we say do? We just got the last three letters from aldaric. I mean the last four letters from aldaric, and then got glucose. We removed the last three letters. We combined that. It formed glucaric acid. Okay. So now glucaric acid, because the functional groups have both been used up. Look at this. The carbon, I mean the carbon which at the carbonyl group or the anomeric carbon has been oxidized. So this one cannot form a cyclic ring because you don't have that functional group to be able to form the cyclic ring. Glucaric acid are going to be used as a chelating agent. A chelating agent is an agent that like will attach to metals. So this one like binds calcium and magnesium. Okay, that's a chelating agent. So the next derivative that we'll be able to talk about is known as an aldehyde. Now, aldehydes, all these we've been talking about were oxidation reactions. But for you to form an aldehyde, what you do is that you get your monosaccharide, you reduce it, it undergoes reduction. Now you can see that the carbonyl group underwent a reduction, right? Okay, there's a reduction that happened. Now, what is reduction? We defined oxidation as the gain of oxygen or the loss of hydrogen. But in this case, what are we defining? Reduction is going to be the gain of an hydrogen. You can see we have gained an hydrogen here. Okay, so that by definition is reduction because that hydrogen wasn't there. Okay, so for example, glucose can be reduced to sorbitol. So sorbitol is a reduced form of glucose. Please take note, huh? Not oxidized but reduced. Aldehydes are formed by reduction, and the way they bring biochemists is they can say glucose is oxidized to sorbitol. True or false? You be quick. Yes, that's true. It's not oxidized; it is reduced. Okay, so you need to make sure that you understand each statement being mentioned. So sorbitol can be used in the food industry as a thickener and sweetener. It is also used as a sugar substitute for patients with diabetes, okay, to substitute the sugar. Now, the other type of these derivatives is a deoxy sugar. This one you already know. Deoxy sugar is where the hydroxyl group is going to be reduced to a hydrogen atom. Okay, so in other words, you lose an oxygen. So for example, at carbon number 2 of this guy, which is a pentose, a ribose, you remove that one hydrogen, one oxygen, you just remain with the hydrogen, so it becomes the deoxyribose. Deoxy sugars are very important in the formation of purines and pyrimidines. We are going to also talk about, so everything we are mentioning here, they will be discussed more in detail. So we'll discuss how purines and pyrimidines are formed. How do you start forming them? These are high yield. Okay. The other ones are known as amino sugars. Amino sugars. Now, when we are talking about amino sugars, what are we saying really? Okay, let me just pause for a bit. I think I've spoken too fast. Are we good up to here? Are we good? Okay. All right. Amino sugars. Now, amino sugars, for these guys, what happens is that you are going to get the OH at carbon number 2 of, uh, let's say, glucose. Then you substitute it with an amine, okay? An amino group. And when we're talking about an amino group, what we are basically talking about is NH2. So this is going to be able to convert our glucose into an amine. So when you add an amine at carbon number 2, we remove the OH. What we've done is we have converted glucose to glucosamine. So in other words, just the same word, glucose. Okay, we're supposed to say glucose. Just remove the E at the end and then get the word amine you put there. So glucose to glucosamine. That is what you can see there. Now, mostly what happens is that when you substitute the glucose with the amine, immediately the amine is going to also add to itself an acetic group or an acetate. So it becomes, because we are adding the acetate to the nitrogen of the amine, it becomes N-acetyl glucosamine. N-acetyl glucosamine. Now, this structure is very important. The N-glucosamine, N-acetylglucosamine is very important in that it is found in structures of bacteria cell wall. So the cell wall of the bacteria is going to be made up of N-acetylglucosamine. Hope that is okay. Okay. Are we good so far? Before we move on further. Okay. Let me just see what's happening to my screen. There's a hand, please. You can go on. Okay. Okay. Let me just do something to it. Seems it's not showing. Are you now able to see? Okay. So the next form of a sugar derivative we want to talk about is sialic acids. Now, sialic acids, these guys have got 9 carbons. Okay, so these are 9 carbon derivatives. And these ones are going to be present on the terminal residue or at the end of glycans. If you talk of glycans, these are basically like polysaccharides, okay, and glycoconjugates. We are going to understand glycoconjugates. Glycoconjugates include things like glycoprotein, glycolipid, those things. So important example of sialic acid is any acetyluramic acid. So what happens is that any acetyl manosamine. Now, if we say any acetyl, we've already talked about how we form any acetyl glucosamine. It means we had used glucose, but when we used mannose to add first an amino group, and then the amino group we added an acetate, it becomes N-acetyl manosamine. So this N-acetyl manosamine is going to be able to react with what is known as phosphoenolpyruvate. Now, this is thing with medicine. Something will not make sense here, but all these two words you are seeing, phosphoenolpyruvate, they might look tricky. All these will be your friends very soon. Okay. So we are going to react N-acetylmanosamine with the phosphoenolpyruvate, and then we are going to be able to form this N-acetyluramic acid. So N-acetyluramic acid and its derivatives are now collectively called sialic acids. What we mean by its derivative is maybe for the same N-acetyluramic acid is we can remove one atom and substitute with another. It becomes a derivative of it. Now, the way biochemistry is they can just say any acetylmuramic acid is formed by a reaction of dash and dash. So you really need to understand each statement. Okay, so that is biochem. So what are the function of sialic acids? These ones are going to play an important role in communication in the cells, also to stabilize the cell membrane, and also binding transport ions and drugs. So we'll discuss this topic in the next class in anatomy. This is what we'll be talking about. On the cell membrane, for you to transport, let's say, calcium from the outside of the cell to the inside or vice versa, you need ion channels. So this is what we are saying. These are going to help in binding, keeping those transporting ions, okay, to the cell membrane. So they help in binding and transporting ions and drugs. Apart from that, they are also used in the formation of proteins like enzyme. We know that enzymes are basically proteins. So they also protect molecules and cells from attack by proteases and glycosides. A protease is something that will break down a protein, and then glycosides will break down a carbohydrate. So this will be the structure. This thing is supposed to have 9 carbons. So what I just needed you to see is just the structure, but you don't need to master it anyway. That is how it looks like. So those were monosaccharides that we've just discussed. Now disaccharides. Disaccharides are condensation products of monosaccharides. These things are easy, right? Yeah, you know them. And this is how it came also last year's exam. Sucrose is made up of what? Just like that. So there's nothing too easy. So other things you see them to be too easy, but other things you just but you are good that you are here. Okay. So table sugar, which is sucrose. We know that sucrose is made by a reaction of glucose and fructose. Now that table sugar, sucrose, when you add it into water, you form what is known as invert sugar. Okay, just take note of that. What is invert sugar? Is basically sucrose and water. Okay, reducing sugars are going to have a free aldehyde or a keto group, meaning that for sucrose, because it is a sucrose is formed by reaction of carbon number 1 of glucose and carbon number 2 of fructose. Carbon number 2 of fructose is the one which contains the functional group. Carbon number 1 of glucose is the one which contains the functional group. It means that both functional groups have reacted. So because of that, when you're talking about this sucrose, it is not a reducing sugar because there's no free functional group. But for maltose and lactose, these two guys are reducing sugars because they are going to have a free functional group. And look at this, for example, when you get alpha D glucose with a beta D glucose, you react them, you are going to form your maltose. And specifically for carbon number 4 of one glucose, it is just donating an hydrogen. Carbon number 1 of the other glucose is donating OH. So that's the difference. Okay. Where does the OH come from? Is carbon number 1 or the anomeric carbon. And then where does the H come from? Carbon number 4. All right. So when you're reacting glucose to form a bond, because it is a bond in a carbohydrate, any bond in a carbohydrate or between carbohydrates is known as a glycosidic bond. So that is the bond that holds monosaccharides together. Now, sorry, not monosaccharides, but disaccharides. Okay. Disaccharides, you can also get to break them down when you react them with water, and it will give you back your monosaccharides. Okay. To be able to break down maltose, you need to use the enzyme known as maltase. So when you're talking about the disaccharide lactose we know it gives you glucose and galactose when you hydrolyze it, and lactose is also known as milk sugar, is found in milk. So this lactose can be broken down by lactase, just like mal- maltose can be broken down by maltase. Sometimes there can be what is known as lactose intolerance. In lactose intolerance, a person does not have the enzyme lactase which can break down the lactose. So what happens that every time you drink milk, you are going to have you start feeling you are going to have a lot of gas in your bowels. Okay, like you are going to have gas. You can even feel in your stomach gas like that. I mean in your intestines. That is known as the flatulence. And it can also lead to diarrhea. That is because the lactose cannot be broken down. So lactose can be able to accumulate, or it can increase, it can go on and on because it doesn't have a mechanism to be uptaken to be used in the body. Okay. And because of that, when lactose is present in the just in the intestines, it can't be taken by the cells. Lactose will start drawing water from the cells. So water will start moving out of the cells, going to where lactose is in the intestines. And that will lead to a lot of fluid or a lot of water increasing in the small intestine. And that is what leads to diarrhea. Are we good? Okay. So something just to know about sucrose. Sucrose made of glucose and fructose. It is formed by plants and not by animals. Okay, that's something take note, not by animals. Now talking about polysaccharides, you are getting a lot of monosaccharides and putting them together. They are also called glycans. So polysaccharides can either be homopolysaccharides, meaning that they've just been made of one single type of monomer or one type of monosaccharide. Heteropolysaccharides are made up of different types of monosaccharides. Now, these polysaccharides can be used for storage or for structural support or structural element. Those which are used for storage include the starch and glycogen. Starch in plants, glycogen in animals. Those which are used in structures include the cellulose and chitin. Cellulose is going to be found in in plant cell wall, but in chitin is going to be found in exoskeletons. Okay, exoskeleton of insects like grasshoppers. Heteropolysaccharides are going to be able to, hetero meaning these guys are made up of different monosaccharides. These are going to provide extracellular support for organisms of all kinds. Extracellular support for organisms of all kinds, no limitation. Okay, so this is how they look. Homosaccharides, you can see, or homopolysaccharides, they don't have different monosaccharides, only one form. Heteropolysaccharides, they have got different forms. So these guys can either be branched or they can be unbranched. Starting with homopolysaccharides, the homopolysaccharides, most important storage polysaccharides are starch in plants and glycogen in animals. So these guys occur in uh, in the cell as clusters of granules. What we mean by granules is they will look as though it's droplets like that. Okay, so they are droplets. So those are clusters of, but they are like droplets which are together, so they are clusters of granules. So starch and glycogen molecules are heavily hydrated. They've got a lot of water. Because of, why do they have a lot of water? That is because the starch and glycogen, remember they are made of glucose, right? And glucose has got a lot of OH functional groups. And what happens that when a molecule has got OH bond or C, a carbon bonded to oxygen, that bond is polar. And because of that, there can be hydrogen bonds happening between the oxygen and hydrogens of other water molecules. So those hydrogen bonds are what causes a lot of water to be together with the starch or the glycogen. That is why these molecules are heavily hydrated. Now, starch itself is going to be found in things like tubers, potatoes, in seeds. It's made of amylose and amylopectin. I'll mention again, there is no statement to undermine in biochemistry. Okay? I think this one came, they just said starch is made up of amylose and amylopectin. You get your marks as easy as that. Amylose is unbranched. It is just straight. Okay, just straight like that. So meaning it only has alpha 1,4 glycosidic bonds, because alpha 1,4 glycosidic bonds are the bonds which make a molecule to be linear. Amylopectin is highly branched. The meaning of that, it has alpha 1,4 glycosidic bonds for to be straight, and then alpha 1,6 which makes it bend. So for starch, amylopectin is going to be bent after 24 to 30 residues, meaning after 24 to 30 amino acids, you are going to have a branch. 30 to 24 to 30 a branch, 24 to 30 a branch. So the branches don't just happen like that. Okay. Both amylose and amylopectin can be broken down by alpha amylase. That's the enzyme that can break them. And you can see what can happen. Alpha amylase can break the alpha 1,4 linkage, and that is going to form maltose. Okay. And then you can also form maltotriose. Maltotriose, you can also form alpha dextrin and such. In other words, you're just breaking down your starch like that. So that is our amylose on top. You can see it is not, doesn't have any branch. only has alpha 1 comma 4 glycosidic bonds, and then amylopectin, which has got branches. Now glycogen is more similar to amylopectin. The only difference is that it has got the branches occur more frequent. You can see for it the branches are after 8 to 12 monosaccharides, different from the amylopectin, which was happening from 26 there to 30. Okay, different from that. Glycogen is most abundant because it's found in animals. It's going to be more in liver and skeletal muscles. And the reason for that is in the skeletal muscles, you need to be breaking down this glucose so that you can use it in, e- when you are doing your exercises. You need a lot of glucose to be available for energy production. The liver also does a lot of work in the body. Detoxification, all the food that will come from the intestines will first have to go, all the nutrients which have been absorbed will first have to go to the liver. So it does a lot of work. So if the liver is not functioning well, that is so bad. Okay. So in the liver, the cells of the liver are known as hepatocytes. So glycogen is going to be found as large granules. We already said what granules are. These are just more like droplets like that. So these granules of glycogen, they also contain enzymes for synthesis. So you have got enzymes which can make glycogen in the same granules, in the same droplets, and enzymes which can break down the same glycogen. So depending on the needs of the body, if I'm fasting, glycogen will be broken down to glucose. If I'm just from eating, glucose will be converted to glycogen. And you understand this is very interesting. We'll talk about how that happens. Okay, so that's the structure. Same with amylopectin. Now, heteropolysaccharides, different from homopolysaccharides. These ones are going to be made up of different monomers. The first example talk about is peptidoglycan. Now, peptidoglycan, if you can hear from the word peptidoglycan, meaning that it has got a glycan part and the peptide part. Peptide is representing a protein. So it has got a protein and a carbohydrate. Okay, that is peptidoglycan. Now, this peptidoglycan is very important. It is a component of bacterial cell wall, and it is made up of alternating. It's a heteropolysaccharide of alternating. Look at this: N-acetylglucosamine and N-acetylmuramic acid. I think this thing came in last year's exam. N-acetylglucosamine and N-acetylmuramic acid. They just said peptidoglycan is made up of dash. Complete. Okay? Alternating N-acetylglucosamine and N-acetylmuramic acid. So the linear polysaccharides are going to be able to lie side by side in the cell wall. And then what happens, remember I've said that these are going to be found in the cell of a bacteria. So these are lying side by side, and then there are also some cross linkages to short peptides, like you are going to have some amino acids in them. That's why we're saying peptidoglycan, because it's supposed to have peptide part. The N-acetylglucosamine and the N-acetylmuramic acid, these are all carbohydrates. Now, how about the peptide part? Those are the ones which are calling as cross linkage. You can see this is one linear chain made of alternating. So if this is N-acetylmuramic acid, this will be N-acetylglucosamine, N-acetylmuramic, just like that. And also these ones, you can see they are lying side by side, and they're now connected together by short peptides like that. Okay, peptides basically amino acids. These close linkages, which we can see formed by these peptides, will be able to make the chain strong, and that is going to be able to prevent the cell from swelling and breaking when the water enters the cell. Remember, we are saying these guys are found where? in the bacterial cell wall. So they make sure that the cell is such strong, even if when water goes into the cell, it does not break. But what happens is that in our tears, we have got the enzyme lysozyme. So in case there are some bacteria which goes into our eyes, because you are not supposed to have bacteria in your eyes. Normally we have got bacteria in our skin, we have got bacteria in a lot of body parts. In the respiratory tract, I mean in the GIT, you have got bacteria to prevent, to fight any. So different bacteria, we call it normal flora, the bacteria which is normal for our body, because it helps us. But you don't need any kind of bacteria in the eye, in the blood, and such. So in the eye, if any bacteria goes there, the enzyme lysozyme, which comes from tears, will be able to break the bond which is found between the N-acetylglucosamine and N-acetylmuramic acid. And when that happens, what can happen is that water can diffuse into the bacteria. And if water can diffuse, it means that the cell is now weak, it can burst open. So that is also how the medicine penicillin works. It is an antibiotic. Okay. So what happens is that when you take this penicillin, it is going to prevent the making of these short peptides that you can see, these linkages with these amino acids will not be made. And when those are not made, it will make the cell wall to be weak, and water will be able to enter, and then the cell membrane will be able to burst. And that is how you get to destroy the bacteria and cure the infection. Are we good here? Are we moving together? Okay, so that was peptidoglycan. The other one is called glycosaminoglycan. Now, the way biochemistry is your first test, mostly you are going to write it online. And what happens is that if you write a wrong spelling, you've gotten it wrong. I remember my first test, this was one of the answers, but I wrote glucosaminoglycan instead of glycosaminoglycan. I got it wrong. So this is glyco, not glycosaminoglycan. Glycosaminoglycan, these ones are also going to be made up of N-acetyl glucosamine. So it can either be N-acetyl glucosamine or N-acetyl galactosamine, but the other component will always be uronic acid. For example, glucuronic acid. Okay, it's an example of uronic acid. Hope you've not forgotten. So any one of these, either N-acetyl glucosamine or N-acetyl galactosamine, any one of those can come together with uronic acid, and then what you form is known as glucosamine. Glucosamine is a very important component. We also discuss it in anatomy. So this one is used as a ground substance. Now, if I say ground substance, think of you want to build your house, as I said earlier. You need bricks. You also need your cement. Okay. But you just don't need that. You also need water, right? You also need some wires that you need to be able to put so that the house becomes strong. So those are examples of what we call ground substance. Ground substances are just there to be able to make sure that they stabilize the way cells are interacting and such things. Okay. You understand them when you talk about that in anatomy. So the extracellular matrix is composed of a lot of these glycosaminoglycans. They are the most important part of the ground substance. For now, just take this as it is extracellular matrix. So when you're talking about a tissue, let me introduce you a bit. A tissue, we know that cells make up tissue, tissues make up organs, and that a tissue will be made up of cells, and not only cells alone. What to hold cells together? So you need cells and extracellular matrix. Now the same extracellular matrix you're talking about will be made up of ground substance. Okay, to be made up of ground substances and protein fibers. So those are the two components. You understand this one very well. Now, some examples of these glycosaminoglycans. An example we can talk about is like hyaluronic acid. Hyaluronic acid has got D-glucuronic acid and N-acetylglucosamine. You can see from these two, which we said it can either be N-acetylglucosamine or N-acetylgalactosamine. We have picked N-acetylglucosamine and also glucuronic acid. Remember we said the other component is what? Uronic acid. And uronic acid, an example of uronic acid is glucuronic acid if we oxidized glucose at carbon number 6. So we always like use the D form, not the L form. That's why you can see the D-glucuronic acid. There's nothing to master here. Hope you are now connecting from where we're coming from. Do you have any question up to here? It is good? Okay. So we've already mentioned what hyaluronic acid is. You can also call them as hyaluronates. These are formed by D-glucuronic acid and N-acetylglucosamine alternating. Now these guys are highly viscous. Hope you know what we mean by a solution being viscous. For example, the way glycerin is. That thing is viscous. It's a bit of more like sticky like that. So these form lubricants on synovial joints. An example of where a synovial joint is on your ball and socket joint, on your shoulder, and also on your hip. Okay, those are examples of synovial joints. Also in the eye, you know how the eye looks like. So the vitreous humor of the eye is also able. It also has hyaluronates. These guys are also found in extracellular matrix. Like they're an example of glycosaminoglycan. So definitely they should be part of the extracellular matrix because that's what we've said is the function of the glycosaminoglycan. The other one, the other example is chondroitin sulfate. Now, chondroitin sulfate is made up of N-acetyl galactosamine, and the same, you can see the glucuronic acid is still there. These ones are going to contribute to the tensile strength of cartilage, tendons, ligaments, and also some blood vessels. Why is it that you can stretch a blood vessel but it can't break? The tensile strength is what is responsible for that, and one contributing factor is chondroitin sulfate. You also have dermatan sulfate. Dermatan sulfate is made up of N-acetylgalactosamine, but instead of use the kind of uronic acid we are using here is not glucuronic, but alduronic acid. It's also uronic acid. You can see the way it ends at the end. This one contributes to the permeability of the skin. What we mean by permeability of skin is that ability of for you can touch your skin now, try to pull it. That ability for you to pull the skin and then it to go back. That is the permeability of the skin. And also of the blood vessels and the heart. Okay. We also have keratin sulfate. So one is formed from N-acetylglucosamine and galactose. N-acetylglucosamine and galactose. Now we say supposed to be uronic acid, so most of supposed to be whatsoever you are going to form. If you get galactose and then you form the uronic acid there. So these guys are going to be present in the cornea of the eye, in cartilage, in bones, also in dead air. Every time you think of keratin, please think of hair, nails, those things. You also have heparin. This is a very nice one. We'll discuss it more in physiology. What happens? It's actually something very important. Okay, what happens during blood clotting? It's made up of N-acetylglucosamine and glucuronic acid or hyaluronic acid. Heparin, this heparin we're talking about is a natural anticoagulant. What that means is that coagulation is the coming together of blood. So this one naturally prevents the coming together of blood. When you hurt yourself, you need blood to come together so that the wound can heal. Because if blood does not come together, blood will continue to pour and pour and pour and pour. At the end, you are going to have little blood and you become anemic. Okay. So heparin is an anticoagulant, meaning that if you've hurt yourself and then you have got a lot of heparin, you will not have blood clotting. But heparin is important within your blood, within your body, because you don't want blood to start coming together. Imagine you are just seated, blood starts coming together, it starts connecting, coagulating, it starts sticking. What will happen is that blood will stop to flow, you end up dying just like that. Okay. So that is why it is important. It prevents blood coagulation or blood clotting. It binds to a protein which is called antithrombin. As I said, all these things will make sense. You remember the word antithrombin. Okay. I think the last part will be now on the glycoconjugates. So we've talked about polysaccharides. Now glycoconjugates. These are just anyway. They are also polysaccharides. Okay. What happens that you are just getting one form of something, you are putting another form. You understand? Examples, you get a protein with a polysaccharide, you form a proteoglycan or glycoprotein or glycolipid. So there are three you need to understand: proteoglycan, glycoprotein, and glycolipid. So those are the glycoconjugates we talk about. These ones are going to be information carriers in the body to be able, in other ways, they are involved in communication between cells and the environment. So they'll be able to label proteins which are destined for transport or for destruction. So if a glycoprotein does not recognize a protein, to mean that this protein does not belong to me, destroy it. Okay, so that is it. So the first one of these is known as proteoglycan. Now proteoglycan, these are macromolecules on the cell surface of extracellular matrix. The components is what is important. Proteo, you can even hear from the word proteoglycan. So one is made up of a protein and the glycan part, which is the carbohydrate. So the glycan part, to be specific, is the same glycosaminoglycan. Please take note of that, glycosaminoglycan. And then you are going to also have now a protein. So those are the two components, a protein and the carbohydrate. The specific carbohydrate is the glycosaminoglycan. So this is how it is. You can see you have got the protein, I mean the glycosaminoglycan, hyaluronic acid on the middle here as a backbone. And then to this now you are having some proteins coming out. These are proteins you call coth proteins. And then to these proteins again you're having glycosaminoglycans coming out. Like for example, keratin sulfate, chondroitin sulfate. Remember we said all these are types of glycosaminoglycans. Also hyaluronic acid is a type of glycosaminoglycan. Okay. So the glycosaminoglycan itself, when you're talking about this proteoglycan, is the main site for biological activity. Even if you have got a protein, it's not the main portion where the activity of this glycoconjugate is going to be happening. So proteoglycans are very important connective tissue, and they are the major component of connective tissue in cartilage. Cartilage is a type of connective tissue. These ones are going to be the major component of connective tissue, such as cartilage. The other ones is the glycoprotein. Now, what is the difference? Proteoglycan, protein, and the carbohydrate. Glycoprotein carbohydrate and protein. What's the difference? So glycoprotein, the only difference is that the kind of carbohydrate that used in a glycoprotein is an oligopolysaccharide. Oligopolysaccharide is the polysaccharide having like 2 to 10 monosaccharides. But for this other one, the protoglycan, the type of carbohydrate we are using is glycosaminoglycan. But it also has a protein. The only difference is on the carbohydrate. We are using oligosaccharide, we are using glycosaminoglycan. So these guys are going to be found in specific organelles. For example, the Golgi complex or the Golgi apparatus, in some secretory granules, and also lysosomes. Okay. The oligosaccharide portion is going to be specific. It will form the specific site for recognition and for binding of other proteins. So this is what you are able to see on this structure. You have got a protein, and then you have got the oligosaccharide end. So we call this protein as an integral protein. And the meaning of integral is that it will pass the cell membrane. It starts from like the outside to the inside of the cell membrane. So many proteins which are going to be secreted in eukaryotes. For example, antibodies, sex hormones like some luteinizing hormones. These are steroid hormone, luteinizing hormone, follicle stimulating hormone. These all are glycoproteins, meaning that they are made up of glucose and they are made up of oligosaccharide and the protein. The other one of importance is a glycolipid. Now, glycolipids are membrane lipids in which the hydrophilic head group, what do we mean by that? These are glycolipids, remember. So there's glucose and there is a carbohydrate and a lipid. So the head is made up of oligosaccharide, and that is the one which loves water. The head loves water. That is why we are saying it's hydrophilic. Okay. Examples are gangliosides. Gangliosides are membrane lipids which are made up of sialic acid and glycosphingolipid. So look at this, for example, a glycolipid here. You have got the lipid, which is the lipid bilayer, and then you have got a sugar on top. So it forms a glycolipid altogether. You also have what is known as a lipopolysaccharides. Lipopolysaccharides, these ones is also lipid and polysaccharide. These ones are going to be found on the outer membrane of bacteria, specifically gram positive. Please take note, okay, for the sake of immunology. These are some questions which come. Gram positive have got a lipopolysaccharide on the outer membrane. You understand this as we talk about immunology. So these lipopolysaccharides are going to be important because they are going to be a target of antibodies. What happens that for the negative, this is actually gram negative. I don't know if I said gram positive. Gram negative bacteria, what happens that they've got these lipopolysaccharides, and because of these lipopolysaccharides, when you take, let's say, if antibodies, because immediately something foreign enters your body, what happens that antibodies are going to be raised to fight it because it doesn't belong to the body. Okay. So those antibodies target the same lipopolysaccharides in order for them to produce a response. So that is what happens. Okay, and we'll end here. The qualitative analysis, we'll talk about it in the next class.